Hello and good evening, dear students. How are you all? I welcome you to the class of biology. Dear students, please report your attendance and we'll begin right away. Hello Itanagar students, how are you? And hello Shillong students, how are you all? Centers, please report your attendance and we'll be starting right away. So from the last part, because I wanted to say this, we have almost completed the most typical unit of biology in class 12, that is genetics. We are about to finish it. So, everyone please report your attendance. Alright. So let's begin. Today we have to start the new chapter, biotechnology. Okay, thank you, Shillong. Itanagar, please also report. Biotechnology. Okay, thank you. And its applications. So basically we have studied about how biotechnology principles are used to make new discoveries, new products. Now we have to study these products. Up till now we have studied the techniques to make a new organism with modified DNA. Now we are going to study the application of those things. So basically, let's write down. Biotechnology is involved in three critical areas of research. What are these areas? Number one, providing the best catalyst in the form of improved organism okay, usually a microbe or pure enzyme. We have to prepare a microbe, or we should have that particular enzyme which belongs to that microorganism. Microbe or pure enzyme. Okay. Also, creating optimal conditions through engineering for a catalyst to act. Third thing is downstream processing techniques or technology to purify the protein or organic compound. the protein or organic compound. Okay, that is the thing. All right, so moving on. After this, we need to define what are the particular effects of seeing these modified organisms in agriculture. So first of all, we will study in agriculture, then we will move on to animals and plants and all other things that are beside the coverage of just the agricultural part. So let's continue. Biotechnological applications in agriculture. Biotechnological applications in agriculture. Applications in agriculture. In agriculture. So how do we describe it? What are the effects that we have seen in this regard? We have to discuss this. So for increasing food production, there are three approaches. First of all, you use agrochemicals. Second approach is use organic agriculture. Use organic products only. Do not use any inorganic products. The third thing is we do not want our yields to suffer. So in the third option, we can take genetically enhanced plants or biotechnologically engineered plants. So let's write. There are three approaches for increasing for the food production at present. At present, there are three approaches to increase food production and what are these approaches? Number one, agrochemical based agriculture. Agrochemical based agriculture. Second is organic agriculture. Organic agriculture. Third is genetically engineered crop based agriculture. Genetically engineered crop based agriculture. Okay. So we all know that there was a great movement in our country called the Green Revolution. It increased the food production in India by about threefold, 300% increase in the food production in India before and after. Okay. Compared to before, we have about 3% more production of food. But considering that there is a ever increasing population growth, it is not going to be enough. Although Green Revolution increased the food production of India increased the food production in India by 300%. By 300%, it is not enough considering the ever increasing population of India. Ever increasing population of India. Right? So therefore we need improved, more and more new improved crop varieties. Therefore we need more and more improved crop varieties. Improved crop variety. So how are you going to grow these crops? This has to be discussed in the previous chapter. But how we'll select them? But how we will select them is another story altogether. So we have to be able to select these crops. So let us start studying about how we have made use of these altered varieties. So you can write, we have been able to modify the genomes of plants, fungi, bacteria, animals, etc. Those organisms whose genome has been modified, those genome has been modified are called genetically modified organisms. Yeah, sure. You can scroll up. Are called genetically modified organisms or GMOs. genetically modified organisms. Or GMOs. So GM crops have better productivity, more crop productivity along with insect pest resistance, so on. Okay. So what are the use of genetically modified crops? How they helped us? GM crops or GM plants GM crops or GM plants, genetically modified crops or genetically modified plants, have helped us in various ways, such as various ways such as various ways such as Churachandpur students, I would ask you to report attendance also because you haven't done that. Okay. All right. Okay. Tilangam, yeah, St. Paul's, you also report your attendance. You haven't done that so far. Okay. So, stress resistant plants. We have been able to make stress resistant plants. Stress resistant plants. What are stress resistant plants? These are tolerant to heat, draft and cold. Heat, draft, cold, salt, etc. Meaning that they are more stable than the others. Other normal human beings, they are more stable. Okay. Reduced consumption of reduces consumption of chemical pesticides. Reduced consumption of chemical pesticides. Chemical pesticides. Reduces consumption of chemical pesticides as plants are able to absorb better. Also, it helps in decreasing the water harvest. Churachandpur. So, what will be the portion for the test? You mean the zoology test or the botany test? I'll let you know. I'll let you know. Okay. Reduces consumption of chemical pesticides. Okay. Increases efficiency of helps in decreasing the post-harvest losses. Post-harvest losses. Post-harvest losses. Increases efficiency of mineral usage. Increases efficiency of mineral usage. Okay. Last point is increases the nutritional value of seed or plant. Increases nutritional value of food. Nutritional okay. So increases nutritional value of food. So basically what does this mean? This means that the vitamins content, the minerals content, all these contents will increase. Okay. Increases the mineral and nutrition offered by the seed compared to before. Increases nutrition value of food. Okay. So let's continue with some very famous examples of GM crops. Some part we had done yesterday in the last class, sorry. Some part we'll do now. Some famous GM crops. We'll try to study them and understand whether GM crops are good or bad for us. So you can say here genetically modified crops. You can say BT cotton. BT cotton. Now what is BT cotton? Let me explain to you. There is a bacteria which produces a toxin, and this toxin is not toxic to human beings, but it is very toxic to insects. So if we can express this gene in cotton plants, what will it mean? The roots of cotton, the stem, leaves, all organs of cotton are protected by the help of this substance. So please write Bacillus thuringiensis, Bacillus thuringiensis is a bacteria Bacillus thuringiensis is a bacteria which secretes a poisonous crystalline protein. Which secretes a powerful poisonous or toxic protein called cryoprotein. Called cryoprotein. Okay. So basically, there is a difference between when you try to spell it up. The difference is, let me go back to the previous slide. Okay. We try to isolate the gene. We try to isolate the gene isolate the gene isolate the gene by allowing only only the non-disease causing or the non-pathogenic part non-genetic non-pathogenic allowing only the non-pathogenic part to remain inside the plasmid. So what will happen if you burn it? To remain inside it. So please write down quickly. Please write down quickly. Cryogene is encoded by Cry1 AC, Cry1 2 AC, AB gene, but that is for another part. Right? So please write. specific Bt toxin gene. Specific Bt toxin genes. Specific Bt toxin genes were inserted into the bacterium into the plant. As a result, the whole plant body produces cryoprotein. The whole As a result, the whole plant started producing Bt toxin. Now, what does this mean? Inserted into the cotton plant, Gossypium, the botanical name for cotton. Now, whenever a plant starts to eat any portion of the cotton plant, it will ingest the toxins also, and when it ingests those toxins, it is going to lead into the death of that. Insect. So you can write here: the whole plant is producing Bt toxin. If any insect consumes a portion of plant body, consumes a portion of consumes a portion of the plant, they effectively ingest the toxin. They also ingest Bt toxin. Bt toxin interferes with the production and metabolism of energy substances, so it is very important. This results in death of the plant immediately. this results in death of insect immediately due to this results in consuming a portion of that thread that is involved here. This results in reduced male population, reduced male population, reduced male, sorry, not just male, reduced, wait a second, this results in the death of plant consuming insects, plant consuming insects, and as a result the plants which are causing harm to the cotton plant, they will not survive, they will not survive. Okay. Cotton is maximum affected by a insect called cotton bollworm. Example, cotton bollworm. Cotton bollworm. Okay. So cotton ball is destroyed by bollworms. So we basically want to remove this insect. It causes a lot of harm. Cotton bollworms are serious pests. Okay. Now there is a issue that is being explored. Okay. This toxin is non-toxic to humans. Non-toxic to humans. This toxin is non-toxic to humans, but lethal to insects. But lethal to insects. Okay. So this is how cotton bollworm is, how we get rid of cotton bollworm. Now there are different types of Bt toxin, even right. There are different types of Bt toxins. There are different type of Bt toxins, and most of the toxins are insect group specific. They are insect group specific. Insect group specific. So each Bt toxin is for a different category of insects. Different types of cryogens are different types of Bt toxins are different types of Bt toxins are effective against different insects. Against different insects. Against different pests. We want to destroy the pests that are eating up our food crops. Okay. So this is about cry-gene. Also, there is another type of cry-gene, like cry-1AC, cry-2AB. Different types of cry-genes are, for example, different types of cry-genes are as follows. Cry-1AC and cry-2AB. Cry-1AC and cry-2AB. Cry-2AB. A, capital B. Now these genes are introduced, we try to decide which gene will be the best, and we run some tests. Based on that, we are doing this. Okay. For example, these are effective against cotton bollworm. Cotton bollworms. But there is another type of cry gene called cry-1AB. Cry-1AB controls corn borer. Okay. So this controls something else, this controls something else, meaning they are insect group specific. Each group is specific to its own type. Also, there is a very new technique called RNA interference technology, and this is having something else. So have you written it down? Have you written it down? Please let me know. As soon as you written it down, please let me know. Okay. So let's move on. So let's move on. We want to cover pest resistant plants. Pest resistant plants. Pest resistant plants. This is something else. Previously it was something else. Why is it so different? The reason is, previously we were using a gene for something so poisonous to insects that they were dying because of it. Okay. Now we are going to use something else, and that is called RNA interference technology. The technique is called RNA interference technology. So plant is going to make some double-stranded RNA, and because of that a particular gene is silenced. So you can say the pest-resistant plants are basically made by RNA interference technology. So let us discuss this. Pest-resistant plants are made by the use of RNA interference technology. Made by the use of RNA interference technology. So how do we do it? There are some pests which have something like muscle proteins, something else. So we target their RNA, messenger RNA, and before it can show its effect, we manufacture double-stranded RNA. Okay. So what is this based upon? You can say plant-based double-stranded RNA is able to silence that particular gene. That particular gene. For example, in myeloid gene in cognitia. Myeloid gene in cognitia a nematode. This nematode causes great harm to tobacco crops. Great harm to tobacco crops. So what we are going to do? This gene is introduced in tobacco plants. The genes for making double-stranded DNA the genes for making double-stranded RNA, double-stranded RNA, as this RNA we can store double-stranded RNA as the gene is silenced for making double-stranded RNA. Just a second. Double-stranded RNA are located in the plant, due to which located in the host in the tobacco plant, any pest harming the host or parasite will be reduced in number. Harming the plant will die immediately. This is how we control it. RNA interference technology. And how are you going to insert this gene? With the help of agrobacterium. Gene inserted with the help of agrobacterium. Help of agrobacterium tumefaciens. Okay, this is another technique. All right. So now let's move on to some biotechnological applications of microbes. Biotechnological applications of like in medicinal products. Biotechnological enhanced products or something like that. Applications in medicine. So how do we achieve it? We already know about humulin, human insulin. So how was it made? There was an American firm called Eli Lilly in 1984. It made E. coli as its subject material and carried out experiments on it. and finally was able to make bacteria which makes human insulin, because the human insulin gene was extracted from human cells. Okay, so you can write here, it is a name given to human insulin. It is a name given to human insulin obtained from human insulin isolated by bacteria. Okay, how do you achieve this fact? How do you achieve this? So regarding this, there is a story. What you have done is, you take two different types of product and raw material. So what we are going to do is, made by Eli Lilly. So you can say here, what did they do? They inserted an inert portion in between, and on the two ends of it, like this, like this. Okay, wait a bit. And after this you have to basically remove the peptide, wait a second, and obtain the purified extract. Purified extract. Just a second. Okay. All right. So what we have here, there is something called proinsulin. And proinsulin is made by joining two parts of peptide A, peptide B, peptide C. So we will consider that later. Okay. All right. So this thing is called proinsulin. You already know about it. Let me just finish this quickly. How are these things being made? Look, there are three portions in insulin. Actually, when we make insulin, we have to make insulin from a single insulin gene which is having three parts. Two parts are active, prepared, and active insulin hormone contains only two peptides, 31 amino acid, 20 amino acids. They have to be joined together by disulfide linkages. That will happen on its own. But before insulin is freely available, it is made something in this manner. It is made as a single long molecule, linear molecule, and this is called proinsulin. This proinsulin, this is peptide C. Peptide C. This is peptide A, and this is peptide B. In order to make insulin, you have to join peptide A with peptide B and have to remove peptide C. Okay, so it goes something like this. This is prepared insulin. Peptide C, sorry, peptide B, peptide A joined together, and peptide C is separated. Peptide C separated. It is free. It is going to be digested, thrown away. So Eli Lilly made the proinsulin, modified and converted it into active insulin. Okay. All right. So how did Eli Lilly do it? Let us write. Eli Lilly Eli Lilly, an American company, in 1983, prepared two DNA sequences. Prepared two DNA sequences corresponding to chains A and B. Peptide chains A and B. A and B. Now what they did was they mixed these chains together, made them separately, mixed them two together, and finally they had the prepared final product. Then they inserted these sequences first in E. coli. They inserted these sequences into plasmids of E. coli. To make insulin chains. And later they extracted and joined them together, extracted them and joined them together to make finished human insulin. Joined them together to make human insulin or humulin. Okay, so this is about human insulin. Then we have another biotechnological application which is a very important one. Finish writing this quickly. Then we'll finish gene therapy. I've already told you about gene therapy also. Please finish writing this. All right. So gene therapy is a story of a girl who was suffering from AIDS-like disease. Severe compromised immunodeficiency is the name of that disease. And this girl luckily had parents who were they had a high capacity to spend the money. But this disease was very severe. She had AIDS-like symptoms. She suffered from multiple infections, and at the age of 4 years, they had to put her in a polythene-like bag, and that was the way she could go outside and play outside. So being inside this bag was her whole story. So it was discovered why she has this disease later by the scientists. They found that she has the deficiency of an enzyme called adenosine deaminase. Adenosine deaminase, ADA enzyme was absent, so in her blood. So it was found that the absence of this enzyme is leading to AIDS-like symptoms. For her immune system to function properly, everybody has a good quantity of adenosine deaminase enzyme, but she does not have this gene and does not have this enzyme. So in order to treat her, the doctors took a very revolutionary approach. And what did they do? They took her bone marrow cells. Bone marrow cells were introduced with the help of a retrovirus, okay, to the gene of adenosine deaminase. Take adenosine deaminase gene from any other human being, cut it properly by restriction endonuclease. Take this cut gene, put it inside this retrovirus. This retrovirus will now infect the bone marrow cells, and this is how all the cells of the bone marrow that have been infected will continue producing ADA enzyme for the rest of their life. Are you getting me? Why did I choose bone marrow cells and not any other cell? Because bone marrow cells are immortal cells. If I chose white blood cells for making this change, making ADA enzyme, there will be a problem. White blood cells have a maximum lifespan of 30 to 45 days. So after 45 days, the disease will again return. So that I don't want. I want that this enzyme should be produced by the girl throughout her life. So in that case, there is only one option to make her bone marrow produce this enzyme with this modified cells. So you can write Gene therapy. So what is happening in gene therapy? Gene therapy is a collection of methods that allows correction of a gene defect, that allows correction of a gene defect that has been diagnosed in a child or embryo. Okay. So how do we do this? The first clinical example of gene therapy the name of that girl is unknown. Her identity is kept secret for some reason. So you can say was in 1990 to a 4-year-old girl with adenosine deaminase deficiency. Adenosine deaminase deficiency. Right? This enzyme is essential for the immune system to function properly. Is very essential for the immune system to function properly. So how did we correct this? In absence of this enzyme, in absence of this enzyme, the person, the girl showed AIDS-like symptoms. AIDS-like symptoms. It's not AIDS. So this disease is called severe compromised immunodeficiency. This disease is called SCID. Severe compromised immunodeficiency. And what happened is, in this disease, because of that, her parents were also suffering as well. That parents did not have that disease. It's a very, very recessive condition, recessive genetic disease. But they were very worried. So, in order to treat this, in order to treat it, we need a cell which can supply lifelong the ADA enzyme. Okay. So her bone marrow cells were isolated. Bone marrow cells were taken out, as bone marrow cells are immortal, and with the help of retrovirus, ADA gene was inserted inside them. Okay. So ADA gene was inserted inside them, and after this, what was happening? After the ADA gene was inserted inside them, these bone marrow cells were introduced back inside her body. These cells were then reintroduced in her body. Her disease was therefore treated. Her disease was treated like this. Okay. So with the help of this information, we have taken out this. So this is all for today. This is all for today, okay? Regarding your test will be for zoology, and I'll let you know. Somebody will communicate regarding the test topics. Okay. So thank you, dear students. Thank you for your time. Travel back home safely, and good night. Thank you.